ammonia. This ammonia, which is majorly produced by the catabolism of the amino acids, and this ammonia that must be in controlled concentration. Its concentration should be controlled in our blood, and the normal range of this ammonia, that is 5 to 35 micromole per liter, and the quantity of this ammonia that should not exceed from this range. If the levels of ammonia or the concentration of ammonia exceeds from this, then this ammonia, this increased level of ammonia, this can be toxic for the body, especially for the central nervous system. And if the levels are not controlled, then there can be several complications, and it can also result in death. So, for this purpose, to control the level of this ammonia, or in other words, you can say that this ammonia is being produced as a result of catabolism of amino acids. So, our body have a system, have a backup system to control the level of ammonia, to control the levels of ammonia, and for this purpose, the body use transamination reactions as well as deamination reactions. With the help of these two reactions, the ammonia levels are controlled. But here the question comes, our main topic is that what are the sources of ammonia in our body except the amino acids, or with the amino acids, what are the other sources of this ammonia which can be lethal, which can be toxic to us. So, there are different sources of ammonia. For example, number 1, the source of ammonia can be glutamine. This ammonia can be produced from the glutamine, as well as this ammonia can be produced by the intestinal bacteria, as well as this ammonia can be produced by the metabolism of amines, as well as this, we can say that this ammonia can be produced by the metabolism of purines as well as pyrimidines. So, we can say that the only amino acids, these are not the source of ammonia, but the other nitrogen containing compounds, they can also be the source of ammonia. Once again, I am going to repeat that this ammonia, this is the end product of the metabolism of amino acids as well as the nitrogen containing compounds. This ammonia is very toxic. This should be in normal ranges, and what is the normal range? That is the 5 to 35 micromole per liter. This is the normal range of ammonia. If the range of ammonia or the levels of ammonia that increases, this can be toxic to us. This can be lethal for the body, and its primary target to affect in the body is the brain, the central nervous system. And if its quantity is not controlled, if its levels are not being controlled, then this can be lethal to us. This can lead toward the death. And in our body, to control the levels of this ammonia, we have transamination reactions as well as deamination reactions, which are usually pronounced as trans-deamination reactions. Trans-deamination reactions. So, with the help of these two reactions, the ammonia is transported in the body to liberate it, and the levels of ammonia, these are controlled with the help of these two reactions. These are the two major reactions. And if we talk about the sources of ammonia other than the amino acids, then its first major source is the glutamine, and other sources are intestinal bacteria by the action of intestinal bacteria, as well as from amines, as well as from the purines or the pyrimidines. First of all, I will talk about the glutamine, that how this glutamine is being involved in the production or in the formation of this ammonia. First of all, you must know that from where this glutamine is coming in our body as a result of this transmyelination reaction as well as deamylination reaction, especially in skeletal muscles. The glutamate is produced, and with the glutamate ammonia is being produced, and when the glutamate plus ammonia, these combines, they form glutamine. So its major source, the major source of glutamine is the catabolism of amino acids, branch chain amino acids in skeletal muscles. In skeletal muscles, whenever the amino acids, they are catabolized in the skeletal muscles, this glutamine is being produced. And what is the glutamine? Glutamine is formed by the glutamate plus ammonia, and after the formation of this glutamine, now this glutamine is going to act as a source of ammonia. This glutamine is taken up by three major organs. Number one, by the liver. Number two, by the kidney, and number three, by the intestine. Now, what is the role of the liver? When the glutamine is taken up by the liver, then what will happen with this glutamine? In liver, there are enzymes. There is an enzyme which we name as glutaminase. So in liver, what we have? In liver, we have an enzyme which is named as glutaminase. This glutaminase remove the ammonia from the glutamine. As I have told you that this glutamine is formed by the condensation of this glutamate as well as ammonia. With the help of this glutaminase enzyme, this ammonia is separated, and we will have glutamate plus ammonia. Now this ammonia is neutralized. We can say this ammonia is controlled, or it is converted into urea by the urea cycle. So this is the fate of glutamine. That is fate of glutamine in the liver. Once again, I am going to repeat that. If we talk about from the glutamine, from where this glutamine is coming, this glutamine is being produced by the catabolism of amino acids, especially particularly in the skeletal muscles. And what is the glutamine? Glutamine is the condensed molecule, is a complex molecule which is a complex amino acid which is formed by the glutamate plus ammonia. And after the formation of this glutamine, now this glutamine, it has a molecule of ammonia, so it can act as a source of ammonia. Now this glutamine, that must be transported to those parts of the body from where this ammonia can be liberated, or from where this ammonia is going to excrete out. Now what are those parts of the body, or the organs of the body? This glutamine, which is produced by the catabolism of amino acid, that comes to the blood. This is taken up by 3 organs: liver, kidney, intestine. If it is taken by the liver, taken up by the liver, in the liver we have a glutaminase enzyme. This glutaminase enzyme will convert this glutamine into glutamate plus ammonia. In other words, we can say that it will separate, it will split the glutamine molecule into 2 parts, the glutamate plus ammonia, from which it was formed. And now this ammonia is converted into the urea by the urea cycle. So as a result, we have controlled the level of ammonia, and here in the liver, glutamine is acting as a source of ammonia for the liver. So this was the fate of glutamine in the liver. Now if we talk about the kidney, what will happen in kidney? In the kidney, for example, suppose this is kidney, ignore the drawing please. Suppose this is the kidney. Now when the glutamine is taken up by the kidney cells, in the kidney cells, this is the glutamine which is taken up by the kidney cells. In the kidney, the kidney also has glutaminase enzyme. And this glutaminase enzyme converts this glutamine into glutamate plus ammonia. Now this ammonia, it will be liberated, or it will be converted into ammoniamine, and this ammoniamine is the major part of acid base balance, or we can say that it is a major part of the buffers. It is helping our body to control the pH, or it is assisting the buffers to control the pH of the blood, pH of the body. Once again, that when the glutamine that is taken up by the kidney, this is kidney, taken up by the kidney, in kidney it has an enzyme which is glutaminase, likewise the liver, and this glutaminase, what it will do, it will split this glutamine enzyme, glutamine amino acid into 2 halves, glutamate plus ammonia, and this ammonia which is going to be produced by the by this reaction, this ammonia will be converted into ammonium ion, and these ammonium ions, number 1, these are involved in the acid base balance to control the pH of the body, and how these are being involved in the acid base balance, that this ammonium ion, it will be liberated or excreted into the urine, and it will control the acidic pH. Is that clear? So we can say that this ammonia is going to play a role in ammonia buffer, So this was the role of kidney, that how this kidney is being involved in the liberation of this ammonia and the excretion of this ammonia, and the source of this ammonia was the glutamine. And the third one is the intestine. In the intestine there are 2 possibilities: either the glutamine is coming from proteinaceous diet, from the protein diet, or the glutamine is coming from the blood, that when the amino acids are catabolized, this glutamine was being produced, and this glutamine is coming through the blood, either to liver or to kidney or to intestine through the blood. So in the intestine, glutamine, it can come from the blood or it can come through diet. Whatever the source of this glutamine is for the ammonia, for the ammonia. Whatever the source of glutamine is in the intestine, the fate or the end result of this glutamine will be the liberation or excretion of this ammonia. What will happen in the intestine? We have glutamate dehydrogenase enzyme or the glutaminase enzyme, which will convert this glutamine into ammonia plus glutamate, and ammonia will be excreted in the feces. Is that clear? So, this was the fate of glutamine in the intestine. So, if the first source of ammonia is a glutamine, then body is going to control the levels of this ammonia by these three, with the help of these three organs, liver, kidney, intestine. Is that clear? So, this was the first source of ammonia. So, in the intestine, this ammonia was being produced from the glutamine and was being neutralized or was being controlled by the help of intestinal glutaminase enzyme. Now, the one point that you must remember, that this glutamine in intestine, only in intestine, the glutamine only in intestine, this can act when this glutamine is metabolized in the intestine, in the intestinal mucosal cells. This can act as these are, we can say that this can produce, this except ammonia, this can also produce alanine as well as citrulline, Is that clear? And this citrulline is further used for the formation of arginine via the urea cycle in the liver, and this alanine, this can be the source, or we can say that this can be gluconeogenic precursor. Once again, I am going to repeat that in the intestine, when the glutamine is taken up by the intestinal cells, we have intestinal glutaminase. There are two sources of the glutamine, either it is coming through the blood or it is taken up by the digestion of diet. Whatever the way of glutamine or whatever the source of glutamine in the intestine is, the end will be the same. What is that? In the intestine, we have glutaminase enzyme that will convert that glutamine into glutamate plus ammonia, and that ammonia will be excreted out. Is that clear? And second is that, or we can say it can be excreted out, or that ammonia will be absorbed through the portal vein, and through the portal vein that ammonia will be transported to the liver, and in the liver that ammonia will be converted into the urea by the urea cycle. So, there can two possibilities, either through the feces, very low amount, or it can be absorbed by the blood, portal vein blood, and from that point it will come into the liver, and inside the liver this ammonia will be converted into the urea. And except this glutamine can also be metabolized to produce the alanine as well as the citrulline. This alanine, if the alanine is being produced, this will act as a gluconeogenic precursor for the gluconeogenesis, and if the citrulline is produced, this will be useful in the conversion or in the formation of arginine, and it can be used in the urea cycle, in the liver. So, this was the first source of ammonia. Now, I will explain you the second source of ammonia. In the intestine, we have urea. This urea is broken down or metabolized by the bacterial enzymes which we name as urease in the intestinal lumen. and this urease will form ammonia from the urea, and this ammonia, it will be absorbed by the portal vein, and from portal vein this ammonia will come to liver, from where this ammonia is converted into again urea and excreted in urine. So this was the second source of ammonia. And then the third source of ammonia is the hormones, monoamines, neurotransmitters, which are protein in nature, hormones which are protein in nature, the monoamines. When these are metabolized by the monoamine oxygenase enzymes, then the ammonia is produced, and that ammonia that must be excreted out from the body to avoid its lethal effects. And the fourth source is purine and pyrimidines, because purines and pyrimidines, they are also nitrogen containing compounds, and when these purines and pyrimidines, these are metabolized, the ammonia is produced, and that ammonia that must be liberated out or excreted out. So at the end we can say that there are four major sources of the ammonia: from the glutamine, from the intestinal bacteria, and from amines, as well as from the purines and pyrimidines. I have explained you that how whenever the glutamine is the source of ammonia, how this ammonia, how the levels of ammonia from this source, they are being controlled. And when the intestinal bacteria, these are the source of ammonia, I have explained you that how the levels of ammonia are being controlled. And when the amines and purines and pyrimidines are the source of ammonia, I have explained you that how they are being involved, or how the levels of ammonia, they are controlled in our body. Is that clear? That's all. That's all about the sources of ammonia.